Good morning, students. We have started about the monohybrid cross, and there we have discussed genotype, phenotype, and after doing the cross by taking only single contrast pair is called the monohybrid cross, and F1 generation, F2 generation, everything we have discussed. Genotypic phenotypic ratio also we have discussed. And from the monohybrid cross, Mendel came to the conclusion and he made the two laws. First law is called law of dominance, and second law is the law of segregation. That also we have discussed. Now next part we are going to discuss about the reciprocal cross. What do you mean by reciprocal cross? No, cross will be made between the two plants and vice versa. Suppose he made the cross between the tall plant and the dwarf plant. Tall plant was the male one and the dwarf plant was the female one. When he made the cross, he got F1 progeny, F1 hybrid, all the plants were tall. Similarly, he made the vice versa. Tall plant made it female plant and dwarf plant made it tall plant. Reversed. At that time also he found out that F1 progeny was the tall plant. So anyway the male and female can be reversed and he got the result. But both the cases he got the F1 progeny were the tall. Now we are going to discuss. In one experiment the tall pea plants were pollinated and a male plant with the pollen grains to a true breeding dwarf plants. The resultant was the all tall plants. Again, when that means here the tall plant was the male. He made the cross with the dwarf plant, he made the female. Okay. Now reversed. When the parental types were reversed, the pollen from a tall plant was used to pollinate the dwarf plant, which gave only the tall plants. The result was the same. All are tall plants. Now, whatever I told, the tall plant, when he made the female plant and dwarf, and another experiment, just reverse. Tall plant, first experiment, tall was made female, second experiment, tall was made the male. But both the cases, he got the F1 progeny, all the plants were tall. Matings were done in both ways, which are called the reciprocal cross. The results of the reciprocal crosses are the same. So it was conclusion from this reciprocal cross he came to the conclusion that the trait or the character is not sex dependent. Sex dependent is not that, but which plant it is present, that is depending on the plant only. The results of the Mendel's monohybrid cross were not sex dependent. That is the conclusion. So one more question. From the reciprocal cross, what conclusion he brought into? He brought into the conclusion that the sex is not determined. Monohybrid, the traits were not the sex dependent characters. Now that is just like 10th standard already. Again I am saying the P means parents. Parents were true breeding tall plant, true breeding dwarf plant, and it is indicated by genotype capital T, small t, because they are true breeding homozygous. Now when made the cross F1 generation, all the checkerboard with it has been told, it has been all are tall. Capital T, two gametes, dwarf plant also two gametes, and the cross has been made. F2 generation, selfing has been done, selfing all the tall plants. So in between tall plants, cross has been made. This is the genotype, this is the female, this is the male. Now again cross has been made. Cross has been made and he got the result. Offsprings of genotypes. Genotypes means we know capital T, one capital T, two capital T small t, and one small t small t. So genotypic ratio is 1:2:1, but the phenotypes are all these two are tall and this is one. So phenotypic ratio will be 3:1. That already we know. The new thing here we are going to discuss dihybrid cross afterwards. The gene for the plant height has two alleles, tall and capital T, small t. Phenotypic and genotypic analysis of the crosses has been shown in the checkup. This one. But you have to study only what do you mean by reciprocal crosses and what is the conclusion of the reciprocal cross. After that, the monohybrid cross. That is 10th standard. Mendel's analytical or empirical approach. Mendel chose to contrast two contrasting characters or traits for each character. So it seemed logical that two distinct factors exist. One is dominant factor, another one is the recessive factor. In F1, the recessive trait and its factors do not disappear. It was there, but it was suppressed, and they were hidden or suppressed or masked. That means suppressed only to reappear in 1/4 of the generation in F2 generation. He concluded that tall and dwarf alleles of F1 heterozygous segregate randomly into gametes. Whatever we have seen, the gamete formation has taken place, and when the F2 generation has been produced, he got 3:1 ratio in F2 generation, the dominant and recessive trait. So he was the first scientist to use this type of quantitative analysis in the biological experiment. Mendel's data concerned with the proportions of offspring. So F1 generation, it was only tall phenotype we have seen, but he told that the dwarf character has not vanished, disappeared, it is masked, masked, masked by the dominant character. Now Mendel's analytical approach is true an outstanding scientific achievement. His meticulous work and precisely executed the breeding experiments proposed the discrete particulate units of heredity are present, and they are transmitted from one generation to the another generation, which nowadays we are calling them as the genes. But he told the factors. Mendel's experiments were well planned to determine the relationship which governed hereditary characters. This rationale is called the empirical approach. Laws that arrived, the two laws have been arrived from this experiment, they are called the empirical laws. That is not an important, but what is the ratio and genotypy, phenotypy, monohybrid. Now next part we are going to discuss about the test cross. Very, very important. Only two things we will be discussing. What do you mean by test cross and what we can find out from the test cross. Test cross is the cross of an individual of unknown genotype with a homozygous recessive. Unknown genotype means when he made the cross capital T, small t, F1 generation he got capital T, small t. He didn't know the genotype, that's why it is called the unknown. But the phenotype he came to know, which is called the tall plant. When a cross is made between the genotype of unknown plant with recessive plant, so he made the capital T small t into small t, recessive parent. This type of cross is called the test cross. So the test cross is the crossing of an individual of unknown phenotype with a homozygous recessive. Understood that one? Very good. Now Mendel's monohybrid cross, all the plants are tall in F1 generation, F2 tall and dwarf plants are, that one we know 3:1 ratio. Mendel pollinated the dwarf F2 plants and got dwarf plants in F2 and F3 generations. So he concluded the genotype of F dwarf was homozygous, and the genotypes of the tall plants are either capital T or small t from the F1 generation. Cannot be predicted. But how can we tell the tall plant is homozygous, heterozygous? To determine the genotype of the tall plant of F1 generation, Mendel crossed the plants from F1 generation. That from here you have to study. To determine the genotype from here, to determine the genotype of the tall plants, Mendel crossed the plants from F1 with the homozygous recessive dwarf. This is called the test. This is the definition. Now, what we can get from this? What is the conclusion? Test cross is used to identify whether an individual is homozygous or heterozygous for the dominant character. That is the use. Reciprocal cross, we have told. Test cross, that is the use. Now, cross has been made. If heterozygous test cross, parental type F1 heterozygous tall plant capital T small t with homozygous dwarf plant, then what ratio we will be getting? 1:1. Tall is to dwarf is equal to 1:1. How? Here two types of gametes, here one type of gamete. So how it will be produced? No, just like cross we used to make capital T small t and capital T small t, here small t small t. So 2:2 it will be capital T small t 2 2. So 2:2. So cross 1:1. Tall is 2. So 1:1 phenotypic ratio. Like that way the cross has been made. Now if the homozygous tall test cross, F1 plant if it is homozygous, capital T, test cross always with the F1 hybrid with double recessive or recessive, homozygous recessive. Now it is one type of plant, it is also one type of gamete will be produced. So all will be tall plants, all the tall plants. So if from the test cross we can come to know whether F1 hybrid is heterozygous or homozygous. If F1 hybrid is heterozygous, then tall is to dwarf is equal to 1:1. If F1 hybrid is homozygous, then all the plants will be tall. All the plants will be tall. All tall plants. That is the conclusion. Now next will be coming, that is called the back cross. Very simple. When the F1 hybrid is crossed with any one of the parents, either tall parents or dwarf parent. Dwarf parents means this is called test cross. So back cross is a cross definition. F1 hybrid with any one of the parental genotypes, may be tall, may be dwarf. The back cross is of two types. How it is? If it is F1 hybrid crossed with tall plant, it is called dominant back cross. And if it is F1 hybrid crossed with dwarf plant, it is called the recessive back cross. It involves the cross between F1 offspring with either of the two parents. Number 3 is when the F1 offsprings are crossed with the dominant parents, all the F2 individuals dominant character, and showed no recessive individuals are obtained. We will show that one. If the F1 hybrid is crossed to recessive parent individuals of both the phenotypes appear in equal proportion, that is 1:1. That is called the test cross. Already we know. Now next one, the recessive back cross helps to identify the heterozygosity of the hybrid. Till here, okay, thank you.